What if I told you your body is full of cells that refuse to die, that poison everything around them, and that scientists have spent a decade trying to kill them with basically no success in humans? Now, what if a team just figured out these so-called zombie cells have been hoarding the ingredients of their own destruction this entire time? A paper dropped in February 2026 that changes how we think about clearing the damaged cells that drive aging. And the weapon? A fatty acid from bitter melon seeds. Let me explain why this matters. Right now, inside your body, there are cells that have stopped dividing but refuse to die. They are called senescent cells, and they have earned the nickname zombie cells for a reason. They are not just sitting there quietly. They are actively leaking a toxic cocktail of inflammatory molecules into the tissue around them. Chemicals that degrade tissue, promote chronic inflammation, and accelerate aging in every neighboring cell they touch. By the time you hit 60, these zombie cells start accumulating fast. Toward the end of life, up to 10% of certain tissues can be riddled with them. And the damage they cause? Cancer, Alzheimer's, heart disease, arthritis, diabetes, pulmonary fibrosis, frailty. Cellular senescence is now officially recognized as one of the 12 hallmarks of aging. These cells are not a side effect of getting old. They are a central engine of it. So of course, scientists wanted to kill them. The proof of concept came from the Mayo Clinic over a decade ago. They engineered mice where you could selectively eliminate cells with a senescence marker called P16. And when they did that in normally aging mice, median lifespan went up by 24 to 27%. The treated mice looked visibly younger, and you did not even have to clear all of them. Removing just 60 to 70% of senescent cells had significant effects. That was the starting gun. In 2015, a team led by James Kirkland identified the first senolytic drug combination, dasatinib plus quercetin. The strategy was logical. Senescent cells survive because they crank up anti-death pathways. Proteins like BCL2 and BCLXL that act like armor plating against apoptosis, the normal programmed cell death pathway. So the plan was simple. Strip away the armor and the cell kills itself. It worked. In mice. In human trials, they showed you could actually reduce senescent cell counts in living people. Over 70 clinical trials of senolytics are now running worldwide. The market is projected to hit $6.4 billion by 2030. Sounds like a success story, right? There is a problem. The biggest senolytic startup, Unity Biotechnology, backed by Jeff Bezos and Peter Thiel, launched with a $700 million valuation. Their lead drug failed phase 2 for osteoarthritis in 2020. The stock crashed 60%. By 2025, the company had ceased operations and liquidated entirely. And the fundamental issue was not just one bad drug, it was the approach itself. Senescent cells have massively upregulated anti-apoptotic machinery. That is their entire survival strategy. They are chronically stressed cells that have built a fortress against death. Trying to force them into apoptosis means hitting them incredibly hard, and when you hit that hard, you damage healthy cells too. Navitoclax, one of the most potent senolytics ever developed, destroys blood platelets so aggressively it limits clinical use. For 10 years, the entire field has been trying to breach the same fortress wall. What if there was a completely different wall nobody was looking at? That is what this new paper found. A team at the University of Minnesota, led by Paul Robbins and Laura Niedernhofer, published a study in the newly launched journal Cell Press Blue. And their central discovery is this. Senescent cells are not just armored against apoptosis. They are quietly accumulating the exact ingredients needed for a completely different kind of cell death. One that has nothing to do with apoptosis at all. It is called ferroptosis. Ferroptosis was formally named in 2012, but the observations behind it go back to the 1950s, when a researcher named Harry Eagle noticed that cells deprived of a specific amino acid died in a way nobody could explain. Decades later, Brent Stockwell at Columbia University stumbled onto it again around 2001. He had a compound called erastin that killed cancer cells, but the death did not match any known pathway. The cells swelled up until their membranes stretched, leaked, and basically exploded. When he tried to publish, colleagues told him cell death was already thoroughly understood, and looking for new versions was outlandish. It took over a decade to prove them wrong. Today, more than 22,000 papers have been published on ferroptosis. The mechanism is elegant and brutal. Iron, which every cell needs in small amounts, catalyzes radical reactions that rip through the polyunsaturated fatty acids sitting in your cell membranes. Normally, an enzyme called GPX4 keeps this in check. It neutralizes the toxic lipid peroxides before they cascade. But when GPX4 gets overwhelmed, the peroxidation spreads through the membrane like fire through dry grass. The membrane ruptures, the cell dies. Now here is what makes senescent cells special. They have been unknowingly preparing for ferroptosis for years. The Minnesota team measured it directly. Senescent cells accumulate dangerous levels of free iron. Not the safely stored kind, the reactive kind, ferrous iron, the form that catalyzes the exact radical reactions that drive ferroptosis. On top of that, their mitochondria are dysfunctional and spewing out excess reactive oxygen species. They are already in a state of oxidative stress before you even touch them, and it gets worse. Senescent cells overexpress the exact enzymes you would need to execute ferroptosis. ALOX15, which oxidizes membrane lipids into lethal hydroperoxides. A CSL4 and LPCAT3, which load polyunsaturated fatty acids into membranes where they become vulnerable to peroxidation. The zombie cells have been stockpiling iron, reactive oxygen, and oxidizable fats like a survivalist hoarding ammunition in a wooden shed. All they need is a match. The match turned out to be alpha-eleosteric acid methyl ester, or alpha-ESA-ME, a modified version of a conjugated fatty acid found naturally in bitter melon seeds and tung oil. The team screened a library of different fatty acids against senescent cells and healthy cells. Most did nothing special. Standard omega-3 fatty acids like EPA and DHA, the ones in your fish oil supplements, barely any effect. But alpha-ESA-ME was dramatically different. The selectivity index, how much more effectively it kills senescent cells compared to healthy ones, was 470. That means it kills zombie cells at concentrations roughly 470 times lower than what harms normal cells. For context, no existing senolytic drug comes anywhere close to that selectivity window, and the proof that this works through ferroptosis and not apoptosis is about as clean as it gets. They threw every type of cell death inhibitor at it. Caspase inhibitors that block apoptosis did nothing. Necroptosis inhibitors, nothing. But ferrostatin 1, which specifically blocks ferroptosis, completely stopped the killing. An iron chelator called deferoxamine, same thing. Leproxstatin 1, another ferroptosis blocker, complete rescue. That is the signature. The lipid senolytics are not breaching the apoptotic fortress at all. They are walking around it entirely, exploiting a vulnerability the cells built into themselves. But why does alpha-ESA-ME work when regular polyunsaturated fats do not? This is the part that I think is going to stick with people. It comes down to chemistry. Alpha-eleosteric acid has conjugated double bonds. That means its double bonds sit right next to each other in the carbon chain instead of being separated. That specific arrangement makes radical chain reactions propagate much more easily through the molecule. Regular omega-3s have their double bonds spaced apart, which limits how aggressively peroxidation can cascade. Alpha-ESA is basically engineered by nature to catch fire once the first radical hits. And there is an additional twist. The team did molecular docking studies and found that alpha-ESA can bind in a specific pocket of ALOX15, the lipoxygenase enzyme that senescent cells overexpress. When it binds there, it appears to make the enzyme more efficient at oxidizing arachidonic acid, the main membrane fatty acid target. So the compound is not just fuel, it is also turning up the blowtorch. But perhaps the most elegant part of the selectivity story is what happens inside healthy cells versus senescent ones. When a healthy cell takes up alpha-ESA, it routes it into triacylglycerol storage, lipid droplets, basically a fireproof safe. When a senescent cell takes up the same molecule, it incorporates it directly into membrane phospholipids, right into the structure that will catch fire. Same molecule, completely different handling. Healthy cells lock the gasoline in the vault. Senescent cells paint their walls with it. The mouse data is where this moves from biochemistry to something potentially meaningful for aging. In naturally aged mice, 20 to 22 months old, which is roughly equivalent to a 60 to 70-year-old human, a short 5-day course of alpha-ESA-Me at 50 milligrams per kilogram reduced senescence markers across the liver, kidneys, and lungs. They also tested it in very old mice, 32 months, and in a progeroid mouse model that ages rapidly due to DNA repair defects. In the progeroid mice, 6 weeks of treatment improved composite health scores. Less tremor, less curvature of the spine, less dystonia. Reduced inflammatory factors in tissues. Now, I need to be straight with you about the limitations, because this is where the difference between an exciting finding and a proven therapy becomes very clear. The sample sizes in some of these mouse experiments were small. 4 mice in the chronic treatment group. That is not enough to make strong statistical claims about anything. The sex of the mice was not reported, which matters because aging and drug responses differ between males and females. There is no full lifespan study in normally aging mice. That is the gold standard the field demands, and it has not been done. The progeroid mouse model, while useful, ages through accelerated DNA damage, not chronological aging. Results in these mice do not always translate to natural aging biology. And critically, while the ferroptosis mechanism was proven beyond doubt in cell culture, the team did not co-administer ferroptosis inhibitors in the living mice. So we are inferring the in vivo mechanism. We have not definitively proven it. There is also a metabolic complication. Alpha-ESA gets rapidly converted to conjugated linoleic acid in the gut and liver. The parent compound has an extremely short half-life in the body. How it manages to clear senescent cells across multiple organs, despite being metabolized that quickly, is a question that needs answering. And the conflicts of interest should be on the table. Robbins and Niederhofer have founded a company called Itasca Therapeutics and filed a provisional patent on lipid senolytics. That does not invalidate the science. Scientists commercializing their discoveries is normal and often necessary, but it means there are financial incentives attached to these results, and independent replication becomes even more important. The journal itself, Cell Press Blue, is brand new, launched in 2026. No impact factor yet. Cell Press is a prestigious publisher, but this paper has not yet been evaluated by the broader field through citations and replication. So is this a breakthrough? It depends on what you mean. If you mean, has someone discovered a completely new way to kill zombie cells that bypasses the exact resistance mechanism that has stalled the entire field for a decade? Yes, that is exactly what happened. Ferroptosis as a senolytic strategy is a genuine conceptual shift. For 10 years, everyone was trying to punch through the apoptotic armor. This team showed you do not need to. The cells have another door, and it is already unlocked. If you mean, can you go eat bitter melon and reverse aging? No. The doses used in mice are high and controlled. The compound gets metabolized rapidly. Eating bitter melon is not senolysis. That is not what the data shows, and it is not what the researchers are claiming. What is actually closest to reaching humans is not this compound specifically, but the broader ferroptosis approach. A company called Rubido Life Sciences dosed its first patient with a different ferroptosis targeting compound in May 2025 in the Netherlands. They are testing it topically for skin conditions first, with systemic trials planned for 2026. Alpha-ESA-Me itself would need formal toxicology studies, manufacturing, and phase 1 trials. That is realistically 5 to 10 years away, if it proceeds at all. But think about what this finding implies at a deeper level. A 2020 study in roundworms showed that iron accumulates, and the antioxidant glutathione depletes over a natural lifespan. Blocking ferroptosis in those worms markedly increased both lifespan and health span. That was the first evidence that ferroptosis is not just a way cells die in disease. It might be a fundamental mechanism of why organisms age at all. If senescent cells are primed for ferroptosis because of their accumulated iron and toxic lipids, and if iron accumulation is itself a feature of aging across species, then maybe the vulnerability this paper found is not just a drug target. Maybe it is a window into something much more basic about what aging actually is at the molecular level. We have spent decades thinking of aging as a problem of cells refusing to die when they should. What if part of it is also cells slowly filling themselves with the chemistry of a death they were never designed to handle? This paper does not answer that question, but it is the kind of finding that makes you ask it. And in science, asking the right question is usually worth more than 100 answers to the wrong one. If you want more of the latest science that is actually changing what we know, there is always something new waiting.